Oh, I think I'm good.
All right, everyone. A couple of announcements before we get started, and I will make sure that we are still actually in the Zoom. Are we still in the Zoom? No, we are not still in the Zoom. All right. So one of the announcements is just to let you know that the link to the YouTube playlist of all our lecture recordings is now up. So hopefully you all saw that message on the announcements slide. If not, recordings are there. They're always there immediately because we are live streaming after class. I will go in and put a title. Maybe I'll clip some of the end and beginning of me fighting with our authentication software. Maybe not. We'll see. But it's there for you to study from. If you missed something or if you missed one of these sort of red words in the guided notes, just skim through those videos, those recordings, and you should be able to get to where you need. Okay, so that's one item of business. Second item of business is that your first lecture quiz will go up immediately after class, or will become available immediately after class. So that's just material that we have gone over in lecture already. And so they're designed to prepare you for the types of questions that you'll see on the exam, but in a slightly less stressful way. So you have it until Monday to complete those. You may use your notes, but they will be timed, and you only get one attempt. So do study before you look at them, but hopefully that'll get you ready for the kind of format that you should expect to see on the exam. Since you have until Monday class time to complete them, the answers will only be available after everyone has completed that quiz. Yeah.
Are tests going to be only on lecture or like the chapters in the book, like stuff that we didn't go over?
Lecture corresponds to what you need to read from the chapters in the book. So like in the part of the chapters where it talks about like organelles and stuff, if we don't talk about it in lecture, you don't need to know it. But if there's a section that talks about all this regulation of enzyme activity, I would recommend looking at that part. Does that make sense? And so you can also go to the outlines in the Google Drive folder. I believe they pull out which chunks you need. So in the future, we're mostly going to go through the whole chapter. It's just at the beginning during the review that we have to kind of pick and choose. Third item of business, not necessarily a big change for you, big change for me. Lindsay, one of our lab instructors and our anatomy lab coordinator, is going to have to take a leave of absence for this semester. So all of you will be seeing me in the anatomy lab. So if you have anatomy lab questions, I'm your girl from now on. But give us a little bit of a grace as we rework kind of covering all of these. So we are going to dive back into enzyme activity. So this is where we left off. We were looking at the fact that we can change the rate of enzymes by changing their shape a little bit. So we do that by binding things on to different places in the enzyme, usually either to a different site somewhere on an enzyme. So I just want to point out when we're looking at the enzyme, we have an active site where the substrate binds in. So that's where our original reactant is going. And then we may have a regulatory site for some other molecule through allosteric regulation. I'm saying this because we're going to be talking about feedback and feed forward in a second. So this could be one of the mechanisms for that feedback or feed forward regulation. The other thing we saw was that we can have covalent regulation of enzymes. So we still have an active site on that enzyme, but this time instead of just sort of fitting in to a regulatory site, we are actually binding on something to that enzyme permanently, covalently, until that bond is broken by some other enzyme. So we had two types of enzymes that might be used as part of this process. So usually the thing that we're covalently bonding on is a phosphate ion of some type. So if we want to add one on, that's a protein kinase. If we want to subtract one, that's a phosphatase. So that brings us to our new content today. Our final bit about enzymes is about feedback. So feedback is going to be kind of similar to our negative and positive feedback loops that we were talking about with homeostasis. It's just specific to a chemical reaction now rather than to homeostasis in the body, which is why we have a different word. But it's the same sort of idea where we're regulating, we're controlling by changing something. Okay, so in feedback inhibition, you can see here, this is a diagram of what that would look like. So we have reactants A, we have this enzyme E1, turns them into this intermediate product B. We have a second enzyme that turns that into this intermediate product C. Enzyme 3 turns it into our final product D. So what we're looking at here, so we see this arrow with a negative sign here, looping back from C to E2, which was that second enzyme. So each of these enzymes was catalyzing a step, so speeding up that step. So what we're looking at in this diagram is we're saying that as we make C, this molecule C, the presence of C slows down this E2 enzyme. So as we make C, it's lowering the rate, that enzymatic rate of E2. So this is an example of feedback inhibition. That's the word for what's happening here. So when we're looking at this map, nothing's changing about that rate for E1. Nothing's changing about that rate for E3. But this rate for E2 is variable because this product that we're making C doubles back and affects that enzyme and affects how well it works. So in this reaction, we would expect this conversion of B to C to be the rate limiting step. So overall, controlling this center part is controlling how fast we make C from A, because this is where we're having the control happen. So we are slowing down this enzyme in the middle as we make more C. So this is ultimately also going to be kind of slowing down these pieces farther along in the process. Question about this. We're going to look at another example of feedback inhibition on the next slide. So a way this might work, for example, is if you think about what enzyme E2 is, maybe C fits into a regulatory site on that enzyme. So that could be a method of creating this feedback inhibition loop. So here's another example of feedback inhibition. Somebody grab that door for a second. Just like shut it. Thanks. All right. So in this example of feedback inhibition, we have the same type of diagram, right? We still have a product that's going back and inhibiting an enzyme. Just in this case, we went a little farther back. So in this case, we went all the way back from our final product, D. And so as we made D at the end, that's going back and turning off or slowing down, lowering the rate of activity of this E1 enzyme. So this is also feedback inhibition. If we have one of our products or something later in the process, doubling back and inhibiting, slowing down an enzyme earlier in the process. It's just now we have a different rate limiting step. So the rate limiting step here, so the variable rate, the one that we're controlling, is this E1 enzyme. Right, so this is how we're controlling the process. This is going to be our rate limiting step, which is why we're affecting this step. Overall questions about feedback inhibition. So feedback inhibition has an opposite, feed forward activation. So this is still going to be affecting the activity of an enzyme, but it's kind of like our positive feedback loops in that it's speeding it up instead of slowing it down. So instead of jumping back and slowing down an earlier step, we can see here that as we make B, that presence of B is speeding up the activity of E3, which we're going to need to convert C to D. So that's just feed forward activation. This is much more clear. So these are just words for like these types of loops, basically. We're talking about those because we are going to move directly next into metabolism. We've looked at metabolism before, so cellular respiration, the production of energy. You know, there are a lot of enzymes involved. We are not going to be talking about each and every one of those enzymes. We're not going to be memorizing enzymes, but it's important to be able to control these steps. This is one of the places where we would see an awful lot of enzymatic activity in the body regulating these kinds of things. So take 1 second, or 2 minutes, I guess, to write down what you remember about metabolism. So what you remember about cellular respiration, what you remember about how we take glucose and make ATP. Okay, so as we move forward with our study of metabolism, I want to just kind of view into what we care most about when we're talking about metabolism and cellular respiration for this course. So if you want to know the absolute most important thing for us to know is what parts of this process are making ATP and how much, right? We want to know where cellular energy is coming from. We care about the steps and where physically in the cell they are happening. And the reason we care about that is that when we start to talk about specific types of tissue, different types of cells, like some have more mitochondria, some might not have any mitochondria. So that's going to make a difference for what parts of metabolism they can do, how fast they can produce ATP, where they're getting their energy from. So those are going to be our two most important things. What parts of this are producing ATP and where are we in the cell? So that when we later look down how cell components vary in different places, we can kind of understand what does that mean for our cell's use of energy and its production of energy. The third kind of little thing would be understanding electron carriers as their main part of this process. So that's our big. So we're going to start by looking at ATP. So we did look at ATP a little bit. What we're trying to do overall in cellular respiration, we're trying to take glucose, so sugar, a carbohydrate that we eat. We're trying to make a form of energy that a cell can use out of that glucose. We don't have molecules, we don't have like membrane proteins that take the glucose and break it down on their own and just use that immediately. We have this sort of currency, right? Like ATP is the cell's money, basically. So we're trying to take this glucose and we're making ATP out of it. The main way we study metabolism, where we always start with understanding how we make ATP, is the process where we get pretty much the most bang for our buck, the one that we see most commonly. So we look at glucose oxidation, sometimes called aerobic respiration, I'd say more commonly called aerobic respiration. So what that is we're breaking down glucose. So that's our first molecule. So our carbohydrate here at the beginning, C6H12O6, that's glucose. So we have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. That is characteristic of carbohydrates, although we didn't focus on that much in our review of biomolecules. Okay. We're going to add in oxygen. Right? We're going to need oxygen if we're doing this full flow that we talk about when we talk about aerobic respiration. And that's important to pay attention to because your cells may or may not have access to enough oxygen at all times. Okay, so they're going to need oxygen in order to do the process we're just talking about. Then we take this glucose and oxygen and we have byproducts. So we have 6 molecules of carbon dioxide, 6 of water, and then the thing we really care about is the energy. And we're going to store this energy in the form of ATP in our cells. And if we were chemists, you would pay attention to probably different things here. So not all of the energy gets trapped as ATP, some gets released as heat. So that can be important potentially in like thermoregulation, like when we talked about shivering, right? Maybe that would be important there. But what we're really focusing on for our purposes is getting ATP so that we can move cells, we can make molecules, we can move molecules across membranes, all really important cellular processes that let our bodies function. So we looked at ATP and we saw it was a kind of modified nucleotide. So we looked at its structure. We saw it was centered around a carbohydrate, a sugar ring. It had a nitrogenous base. And then the part we care about when we're talking about how we make ATP and how we store energy are these 3 phosphate groups. So this triphosphate part. So when we're talking about metabolism and we're talking about storing energy in the form of ATP, what we're going to see is that all those steps in metabolism and aerobic respiration, what we're trying to do is have exergonic reactions, the reactions that release energy. We're pairing them up with this process of converting ADP, adenosine diphosphate, into adenosine triphosphate ATP. So we're using reactions that release energy in order to create this bond here and tack on a phosphate that we'll see later on. We basically snap and unsnap this phosphate on and off as we use energy and store energy throughout cellular life. So when we're synthesizing ATP, we're going to need that starter molecule ADP, right? That's the building block. So that's going to have our sugar, it's going to have the nitrogenous base, and it's going to have the first two phosphate groups. ATP is adenosine triphosphate. So we're going to need another phosphate ion from somewhere. And we're going to need energy in order to form that bond. So creating ATP and some water from ADP and a phosphate ion. This is an endergonic reaction, meaning it requires energy. It needs to soak up energy to form that bond. Okay, so that's why we pair this endergonic creation of ATP with other exergonic reactions that release energy steps. This happens to be a condensation reaction. So as we do this, we give off water. So we produce some H2O in this process. So we could sometimes refer to this reaction that we're doing as a phosphorylation reaction. So phosphorylation, we have our phosphorus in here, right? So phosphorylation, we're making it phosphorus, we're adding on phosphorus, right? So that's talking about the fact that we are adding this phosphorus ion onto the ADP, and that's how this process happens. There are two ways of doing this. So we are going to see that we have substrate level phosphorylation. So now we know what a substrate is, right? Substrate, something binding into our enzymes, right? So in our substrate level phosphorylation, that's the kind of process we're going to see as we make ATP, right? We're basically like transferring a phosphate group from something using some enzymes onto the ATP. We're also going to have a type of phosphorylation called oxidative phosphorylation. And this is specifically going to happen at the end of aerobic cellular respiration. It's going to be our step that requires oxygen. And this is where we're going to see something called the electron transport chain embedded in our inner membrane of a mitochondria. What they use is the energy of electrons in order to tack free phosphate ions onto ADP, giving us ATP. We're going to see that oxidative phosphorylation gives us a lot more bang for our buck. This is going to be where we see most of the ATP made. This is going to be our most effective type of glucose oxidation, our most effective way of getting energy out of glucose. But it doesn't always need to happen because we do have this other way of producing ATP. We do also have this substrate level phosphorylation that's going to happen in some of the earlier steps of our process. So eventually, we're going to see that we will use ATP. We'll break it down to release that energy again. So like when we talk about how muscle cells contract, we're going to see that ATP is going to be needed, and we're going to pull off that phosphate ion to release energy to allow a muscle to contract. So we'll see the breakdown of ATP later in this course as well. What we want to understand really is that that energy is being stored in that final bond with that final phosphate ion on it. So since the creation of ATP is a condensation reaction that produces water as a byproduct, breaking down ATP is a hydrolysis reaction. So this is kind of reversible, it goes both ways. So if we break down ATP, we can do that by adding in water. So we're back to this equation, but we've tacked on some things. So we're still looking at the breakdown of glucose. And we're still talking about producing energy, but now we've specified some numbers. We've specified that here we got about 38 ATP. You'll see this number on how many molecules of ATP we get is variable. That is because it is actually variable in different tissues in your body. It is different between different people, depending how old you are. It's kind of dependent on how leaky membranes in your mitochondria are. So don't get too hung up on this overall number at the end if it varies between your textbook, what we talk about here, or other resources. So we still took our glucose, made ATP. We needed oxygen to do it. And this gives us some idea of the ballpark numbers we might be seeing here for our energy production. So here we see that we put in some ADP, that adenosine diphosphate. We put in some phosphate ions. And together, these two are where we're getting that ATP. I wouldn't focus too much on overall numbers in these equations. The place I would want you to remember numbers is when we talk about like how many ATP does glycolysis make. But in terms of memorizing chemical, in terms of memorizing chemical equations, I'm not going to like make you write down this full equation in the future. So just so you know, since you all have had a little bit of chemistry, because it is a prerequisite for this course, the energy in 38 molecules of ATP, as we saw in this reaction, is about 266 kilocalories. The energy released during glucose oxidation, so that breakdown of glucose, is 686 kilocalories. We needed about 420 here, so this is the difference of energy when we synthesize glucose. Some of that energy, some of that difference there is getting released as body heat. So again, this is a place where I don't expect you to memorize these exact numbers, but I want you to just kind of understand this process. So we are storing some of this energy in ATP, but also we're losing some of it. This is not a perfectly efficient process. Some of this is creating body heat. So we're going to go through the steps now. And depending on what source you look at, you will see different sort of breakdowns of steps occasionally in terms of like which numbers they are, exactly what they call them. So here are going to be our three main ones that go along with our textbook. So glycolysis, and you will always see glycolysis as a step. And that's because this is a step that happens when there's no oxygen also. So it's part of our full process, but it can also happen independently. The Krebs cycle, which is also called the TCA cycle, the citric acid cycle. Then at the end, we're going to have the electron transport chain doing oxidative phosphorylation, essentially. So those are going to be how we're going to kind of break up these steps here. And the reason we're focusing on these are the places that we see ATP production. We see some ATP production in glycolysis. We see some ATP production, especially in oxidative phosphorylation. These are where we're focusing on energy. So glycolysis and the Krebs cycle, or the TCA cycle, the citric acid cycle, those are where we see substrate level phosphorylation. So that's when we're thinking about enzymes and moving a phosphate ion. The electron transport chain is where we see the oxidative phosphorylation, where we're using these electrons that we've been moving around in earlier parts of the process using our electron carriers. We're finally using those electrons to tack on these phosphate ions inside the mitochondria. So I'm going to start with glycolysis. So what's happening in glycolysis is we're going to start at the beginning. So we're starting with glucose, our big 6-carbon molecule, right? So we're going to break that down, and we're basically breaking it in half. So glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. So we just split that glucose in half. Technically, within glycolysis, there are 10 steps. So 10 reactions, each of which is associated with an enzyme. So enzymes are lowering activation energy barriers, helping us get from glucose, our 6-carbon molecule, to this 3-carbon molecule, the pyruvate. This whole process is happening in the cytosol. We don't need to know about each of those 10 enzyme catalyzed reactions for the purposes of this course. You could focus on them if you become an expert in glycolysis, an expert in metabolism. But for us, what we want to know is that we're getting 2 ATP on net out of this process of glycolysis. We're making a little bit of ATP for ourselves. So this is a place where you do want to remember that number, that 2 ATP. We are also getting some electron carriers. We're putting some electrons here onto our electron carrier. This is something we want to focus on. You don't need so much the numbers here, but you do need to understand this process. So we are taking here this NAD plus molecule, and we're reducing it to NADH. And this is important because this NADH is going to go ultimately to our final step, to the oxidative phosphorylation with the electron transport chain inside the mitochondria. So we do need this glycolysis step in part to fuel that reaction at the end. So we're going to see as we move through these steps, we're going to be reducing these electron carriers a lot. We're going to see these reduced electron carriers building up so that we can kind of explode at the end and do this explosive oxidative phosphorylation step and get a lot of ATP. So all of these earlier steps are kind of feeding into that process. We are also focusing on this NAD, NADH step, because when we run glycolysis independently, like we don't go into the mitochondria, we never get to oxidative phosphorylation. That means we're going to have to do something else with this NADH, right? If we can't send it to the end of the process, we're going to have to do something else with it. So we'll see that come back when we start to talk about lactate, basically. Okay. So there's no oxygen here in this process. We're not making any carbon dioxide because carbon dioxide comes when we peel off a carbon molecule, and we just went from a 6-carbon molecule to 2 3-carbon molecules. We didn't lose any carbon yet. We didn't use any oxygen, and this is happening in the cytosol. So when we think about our cell, we think about our intracellular fluid in that cytosol, in that cytoplasm. We're out floating around, still in that intracellular fluid. We are not inside any organelles yet. We are not inside the mitochondria. That is important when we talk about what cells have mitochondria and how many they have. So glycolysis can happen no matter what, because it can happen in the cytosol. It doesn't need to be in that specific organelle. Questions about glycolysis before we move forward? Just a thing that trips people up. I do keep repeating this bit about the fact that glycolysis can happen in the absence of oxygen, and we'll see it later, because that does kind of confuse people sometimes. So that you can almost think of glycolysis as the first step of 2 different processes when we get to our reduction of pyruvate into lactate. So thinking about that NAD and NADH stuff. So I explained it to you in the way that I think about it, in the easiest terms that I can think of in my head. But technically, these are energy transferring nucleotides. So when we were looking at the nucleic acids, we also looked at what these might look like. They are technically coenzymes. So they're going to work together with our enzymes in the electron transport chain in order to help them function. And NADH is the reduced form of NAD. If you remember that mnemonic, Leo the lion says GR, reduced, gained an electron, that's the GR, gained an electron reduction here. So it has the extra electron, so it's carrying this electron on it. So glycolysis made 2 of this, so it made some. We're going to see that processes later on are going to make more than that. Here's the whole process of glycolysis if you are curious about all the enzymes involved, but we do not need to memorize this for this course. But this is kind of look forward potentially into your future, depending where you go. You may focus on this a lot more closely. For our purposes, the one thing I want to highlight here is that at the beginning, when we are breaking down that glucose, we actually have to put some coins in to start, right? We have to start up this process with a little bit of energy. So if you read out chemical formulas for glycolysis, you might see that we're actually putting in some ATP here to jumpstart this breakdown of glucose. So in this energy investment phase, before we can break the glucose in half, we're basically like kind of shaking it up and making it more unstable in order to allow that breakdown to happen. So that's why we're investing energy in this energy investment phase. Then we have our energy harvesting phase. So that's where we get our ATP out, right? So we're going to get some ATP out. And this is how we got to that 2 total number. So the energy harvesting phase makes us 4 ATP. We had to put in 2 ATP at the beginning in order to make the glucose unstable enough to break it down. So if we want to know like how much on net our cell earned, right? Like how useful was this for us? We take that 4 that we get, we subtract our cost, right? And we get that 2 ATP. So that's where we got the 2 ATP on net from in that previous slide. So you should know that that 2 number for glycolysis is net. It's not like every ATP molecule that ever touched these reactions. And more pictures of this whole big complicated process. Take a second, review for yourself. Remind yourself glycolysis is generating ATP via a specific type of reaction. So who thinks A? Who thinks B? Who thinks C? Nobody wants to read that. Who thinks D? Okay, so it is substrate level phosphorylation. So we can key in on these pieces. All right, so we could just remember that glycolysis works via substrate level phosphorylation. But if you want to understand why that is, right, we can look here and we can see that we have an enzyme adding in that phosphate, right? And specifically, we have a phosphoglycerate kinase adding on that phosphate, right? We have another kinase here. We know kinases, right, kind of tack on phosphates, so it makes sense that these might be named like that. That's substrate level phosphorylation. Those are those enzymes involved there. You don't have to remember them. So in your text, the next piece that we're going to talk about doesn't list out that as a numbered step again. So like, don't get too hung up on numbers of steps is kind of what I'm telling you. We have a linking step is how your book likes to think of it. So in this linking step, we're really talking about how we get to the Krebs cycle, which is going to happen inside the mitochondria. So in our linking step, we're taking that pyruvate that's still out in the cytosol, so that's when the glycolysis is happening. We're going to move that ultimately into the mitochondria. And we are also adding on some things to that pyruvate to make it the form that we like for the Krebs cycle. So we are tacking on this CoA piece to make acetyl-CoA from our pyruvate. So the acetyl-CoA came from pyruvate, adding in CoA using an enzyme, pyruvate dehydrogenase, in order to form this acetyl-CoA. In this process, we did reduce a tiny bit of NAD to NADH. So we did make an electron carrier here, still going to that end electron transport chain. We're not using it yet, but now it has an electron full of energy that we're going to want later. But we didn't produce any ATP. So it is a little bit involved sort of vaguely in our energy production because of that association with our final step. But no ATP is being made here. So that's why we're thinking about it as a linking step. That is why we are thinking about the Krebs cycle as sort of our second step, because it's the second step where we see ATP production, right? So if we look at this map, we can see already that we're going to have some ATP. So the Krebs cycle, or the citric acid cycle, the TCA cycle, it's a cycle, it's a circle, it keeps happening over and over again, going around and around. So our reactants, our starting molecules, are also our endpoint, and they can go straight back in so long as we are maintaining appropriate amounts of these molecules that we need. So this is happening in your mitochondria. So if we didn't have mitochondria, we could not do this step. Okay. It's going to produce some ATP, but if we did not have mitochondria, we are not producing that ATP because we are not doing this step. Here, we do have a lot of reduction of coenzymes, by which we mean we're taking a bunch of these electron carriers and putting on a bunch of electrons, sending them to oxidative phosphorylation. We only sent a couple of reduced enzyme carriers. Yeah, we only sent a couple of these NADH molecules from glycolysis and from that linking step with pyruvate, but now we're getting a lot more. So that's kind of the main thing, the most important thing about the Krebs cycle is that it is reducing all these electron carriers. So this is mainly where we're getting electrons for our next step. So the earlier steps do a little bit about that, but it's mainly here that happens. Okay. So our acetyl-CoA from that pyruvate linking step is our initial substrate. So our first molecule that goes in, and then we go around in a cycle. Now, we remember we had our glucose in glycolysis. We split it in half into two pyruvate, pyruvate. And then each of those pyruvate was undergoing this linking step. So ultimately, this one became an acetyl-CoA. And this one is now in an acetyl-CoA. So the reason I'm showing you this is that the Krebs cycle is just showing you what happens to one acetyl-CoA. So anytime you see numbers associated with the Krebs cycle or the citric acid cycle, whatever you call it, you're going to have to double them if we're asking about per molecule of glucose, right? So per molecule of glucose, this wheel spins twice, essentially. Okay. So per each acetyl-CoA, we're ultimately going to make 1 ATP molecule, 3 NADH, 1 of this FADH2, which works basically like NADH. And we are going to peel off some carbon dioxide here, so we're going to lose some carbon from our molecule. Okay. So this means, because I really care, that we understand the ATP, right? So per each acetyl-CoA, we produce 1 ATP, which means that for a molecule of glucose, we made 2 because we went around twice. So to this point, on net, when we broke down glucose, we put in 2 molecules of ATP to shake it up. Then when we broke it in half, we got 4. So we had 2 net ATP from glycolysis for 1 molecule of glucose. We moved pyruvate into the mitochondria. Didn't have anything happen with ATP there. But we did make this acetyl-CoA, which is now going into the Krebs cycle. We're going around twice because we have 2 acetyl-CoA. So we're making per molecule of glucose 2 ATP. So we're up to 4 now if we're tracing per molecule of glucose. Questions about that sort of big picture process? And we can focus on the big picture for our purposes. We do not have to focus on all these little enzymes, but they're pretty cool. We have a number of different steps here, right? So we're going through these 8 steps, each of which has an enzyme associated with it. So this is why we are thinking about this as substrate level phosphorylation again, because we're using these enzymes essentially. So this took us to oxidative phosphorylation. And we start off next time, we will know that through glycolysis and the Krebs cycle, we have 4 ATP molecules. And we're getting ready for the electron transport chain to explode in this oxidative phosphorylation step. And you need oxygen to start a fire, so you're going to need oxygen in oxidative phosphorylation. I will see you on Monday.